Hi, my name is Barera Ajaz from Fordham University.
And I'm Harley from SUNY New Paltz, and this is our presentation on generating a molecular atlas of the non-pregnant female reproductive system. Just as a content warning, the first two slides do have images of actual organs, so if you're squeamish, maybe look away from those.
All right, so let's start with the basics. Below, we have two images of the female reproductive system. One is a diagram and one is the actual image, and we're going to be looking at the diagram on the right. Let's start with the vagina, which is the entry. It is the entryway from the outside world into the female reproductive system. We then go into the muscular cervix, which is the passageway between the vagina and uterus. In the actual image, the cervix is not pictured as the vagina is covering the cervix. From the cervix, we go up to the uterus, which waits for a fertilized egg that it then nourishes and develops into a baby. If no fertilized egg is present, then it sheds the functionalis layer of the endometrium, which is the outermost layer, every month in the menstrual cycle. From the uterus, we go into the two fallopian tubes on either side. The fallopian tubes are passageways between the uterus and ovary and provides a place for the egg to be fertilized by the sperm. From the fallopian tubes, we finally go into the two ovaries. The ovaries function in production, storage, and release of oocytes, which are immature egg cells, and they also function in the secretion of hormones to regulate menstrual cycle and pregnancy.
And then the fallopian tube. So we did work with the other tissues present in the female reproductive system, but we focused on the fallopian tube most heavily. So we're just going to go over that anatomy right now. It can be broken down into four main sections, and you can see those here. Starting on the left, the outermost part is the fimbriae, and this has finger-like projections. And those can be used to pick up the eggs that are released from the ovary and transport them down the tube. The next section is the infundibulum, and that's that curved section there. And then that's followed by the ampulla. The ampulla is the section in the middle. It's the largest part. And then we have the isthmus, and the isthmus connects the tube back to the uterus. One of the big reasons the fallopian tube is so important is because of its connection with ectopic pregnancies. So an ectopic pregnancy is just a pregnancy where the fertilized embryo will implant somewhere else other than the uterus where it's supposed to. It can be very painful and it's very dangerous. These pregnancies do not result in viable births, and if they're left untreated, then they can also be fatal for the pregnant person. 90% of all ectopic pregnancies implant in the fallopian tube, and 80% of those implant in the ampulla. Even when you take into account that it is the largest section of the tube and the longest, it's still disproportionate implantation in the ampulla versus any other section of the tube.
Now that we have the basic anatomy down, let's move on to the project itself. It's taking place at the Penn Tissue Mapping Center at the Kim Lab. So the goals of the Tissue Mapping Center are to use tissues from different samples and donors to create a molecular atlas of the female reproductive system. Using this atlas, we want to examine the differences between regions in the same sample and regions in separate samples. We want to connect these to the function of the region and examine how the differences might be connected to an increased susceptibility to disease or complications. The Penn Tissue Mapping Center wants to provide the atlas to the HuBMAP Common Coordinate Framework, where other atlases are uploaded. This atlas can be used by other researchers to further our knowledge on the female reproductive system.
So far, we do have some data already. So with multi-omic sequencing, we've been able to detect clusters of different cell types. And with the Koya or Codex imaging, we've been able to look at the samples and see the protein expression and spatial relationships between those. We currently have 6 donors ages 18 to 42, and they have various numbers of gestations to really capture the available biological diversity. There are still more data points that we need to collect. Right now, the single cell protocol does not show cell markers that we know should be there, like oocytes. And we are currently optimizing the protocol for data extraction from uterine tissues, which includes both the uterus and cervical samples.
Our goals are aligned with the goals of the Penn Tissue Mapping Center and the data that is already present. We want to continue multi-omic sequencing and focus on optimizing the extraction and isolation protocol for cervical nuclei, since the previous protocol did yield degraded results. With the multi-omic sequencing, we can do ATAC sequencing, which shows us the epigenomics or accessibility of different segments of the genome, or RNA sequencing, which shows us the transcriptome. We also want to continue Akoya imaging, focusing on obtaining accurate staining for different cell types in each section. We are especially hoping to optimize the antibody concentrations present. We will begin doing RNA fish on the fallopian tube. With RNA fish, we will be able to create a spatial image of the sample with the information in the RNA, as opposed to Akoya, which gives us information with proteins. This is optimal because it will allow us to see if there are any errors in either the proteomics or transcriptomics of our samples. We will also begin doing bulk RNA sequencing on the uterus. Bulk RNA sequencing allows us to see the overall RNA expression in the tissue, as opposed to multiome, which gives us a single cell level of expression. This will allow us to potentially detect markers that should be present in the multiome that are absent, for example, the oocyte.
So starting with the nuclei isolation optimization for multiome, we started working with cervical tissue that had been snap frozen, and we did a number of trials, and we found that we got the best results with the manual dissociation. So taking the tissue and cutting it into tiny pieces with a Dremel and a scalpel, and then increasing the time it spent in the lysis buffer. We ran those samples through the bioanalyzer to see how effective each trial was, and those are set to be sequenced. And with this, we can get a better idea of what cell types are present using ATAC and RNA sequencing. And we've already begun to see some different cell types like glandular epithelium and macrophage tissues.
As said before, we continued Akoya imaging and analyzed the images. Akoya imaging is a highly multiplexed immunohistochemistry stain. In this, a primary antibody with an oligoprobe and fluorophore attached binds to a specific protein in the sample. Each protein is specified to a cell type, allowing us to detect different cell types with their protein expression. The imaging has multiple rounds of hybridization stacked, and this just means that one round of probes attach to the antibodies and a picture is taken. The probes then wash off and another round happens with another picture. In the end, the images are stacked together to give a spatial image of the different cell types. In the fimbriae, which is pictured on the right, the fimbriae is a section of the fallopian tube, and we're able to see several different cell types denoted by a protein. For example, we see ecadherin, which is the magenta color, and that denotes an epithelial cell type visible in the image. And we can see in the list next to the image of the fallopian tube that the DAPI protein does not denote a cell type because it's used to show the presence of the nucleus. And this just allows us to see a more accurate view of where each cell is. We were able to optimize most of the antibody concentrations, but we'll further work on optimizing them to another level.
Next, we did single molecule FISH. So FISH is fluorescent in situ hybridization. Basically, we're using fluorescent probes attached to the tissue so we can get a spatial picture of it. We ran the first trial with 4 samples from the fallopian tube, stained with a total of 5 probes, 4 unique and 1 control. We did image those samples, but they did not come out as uniform or brightly as we had anticipated. So we adjusted the protocol with an increased incubation time and re-imaged, and that image you can see on the left, and we got a brighter, more uniform picture. The magenta in that image shows the nuclei, and then the different colors are indicative of different cell types. And we will compare this with the Akoya images to get more information about the proteomics and transcriptomics, and we want to image more samples in the future.
The next thing we worked on was bulk RNA sequencing. As said before, we wanted to do the bulk RNA sequencing to detect the presence of markers in the whole tissue to ensure we were getting positive results in the single cell sequencing. The bulk sequencing process is very long, and so far we have been checking the integrity of our samples. So one worry that we had was that as we process the samples, they would degrade too much and we wouldn't see optimal results. We checked the integrity of the samples by extracting the RNA and checking the RIN score. The RIN score is the RNA integrity number of the sample and is measured on a scale of 1 to 10, 1 being the most degraded and 10 being the least degraded. We were aiming for a score of around 7, and we can see that on the RIN score of the isthmus, which is the top left picture. The isthmus is a section of the fallopian tube, and it has a RIN score of 7.10, meaning medium degradation, which is what we were aiming for. We used a neuron suspension as a positive control, as neurons are collected fresh and have low RNA degradation, as can be seen in its high RIN score of 9.4. As we continue, we hope to use the extracted RNA for bulk sequencing and to use the bulk sequencing data to compare to cell types found in other single cell assays. The last thing we did work on was annotating whole images of tissue with HuBMAP software. In this, we used images of images of sections of the ovary obtained with Visium, a protocol that captures RNA expression and maps it onto an image. With QPath, we were able to annotate certain sections of cells, like the ovarian follicles we can see. Ovarian follicles are just oocytes. We were also able to capture more widely distributed cells with the Segment Anything Meta AI software, aka SAM. With SAM, we were able to capture chunks of similar cell types with the AI-powered segmentation, as opposed to having to do it manually. And we can see an example of the SAM on the picture of the right of the stromal cells. We did the image annotation as it allows us to see the differences in sections of the same tissue, enabling a more accurate image of the tissue as a whole.
So areas for further research, this is still an ongoing project. So we are continuing to build the atlas using the various methods that we mentioned and comparing them together. There are a number of different tissue types and parts of the female reproductive system, so it is a pretty extensive project. We're also focusing on ectopic pregnancies, like I mentioned before, seeing if there are cell types present that will give us more answers as to why embryos implant in certain places. We also want to expand our library of proteins that we test for with proteomics. This will allow us to see a more in-depth version of the tissue and allow us to find more specific cell types and subcell types. Finally, we're hoping to study heterogeneity a little bit more. We want to compare the data we receive from different donors and samples and see whether or not those differences are indicative of underlying disease states or different issues.
We want to thank everyone in our labs for their instruction this summer, with special thanks to our principal investigators, Dr. Jun-Young Kim and Dr. Kate O'Neill.
And we also want to thank the NIH and HuBMAP Consortium for funding and for this opportunity. And a special thanks to Maya for setting everything up.